my name is Rachel. I'm an undergraduate researcher who's pursuing graduate work now. Yay, go me. And I'm going to talk to you a little bit about my atrocious mistakes working with zebrafish brains, which is something that you've probably always been curious about. So really, you're like, why do you care about zebrafish brains? Great question. We are trying to study fear and how fear works. And so this is likely something that you can recognize that in humans, fear behaviors look really, really different. So we have fear of someone breaking into our house, we have fear of scary monsters and fear of spiders. But all of these different stimuli create different fear responses in all of us. So it makes it very, very difficult to study this behavior and quantify it in a meaningful way. So we can extend that and we can begin to study fear in animals. Again, this becomes a little bit more complex with mammals because you can see that we have lots of fear responses. I'm going to see if I could. The audio is not super important to this video if you've ever seen it. This is a cucumber. So the cat's going to turn around and it's just terrified. But look at this response that we see in a dog who's running away from a jar of mayonnaise. So, and then this video proceeds for like 10 minutes of other things that this dog is scared of. So it makes you wonder, is the dog scared of the human or the items? We're not sure. So bottom line is that it can still be very complex to study in animals like this because we have lots of responses to different stimuli. Now is the bottom line that you're really interested. So what about fear behaviors in fish? And this is probably a hypothesis that you have all unwillingly tested at some point, probably at the dentist's office, represented here with our dear friend Darla from Finding Nemo. So right, the continual tapping on the glass, we see almost the same response from all of the fish in the aquarium, that they are running away from, well, fish don't run, but you know what I'm saying. They're moving away from the glass, and they are trying, it's the scatter response, that they almost all have the same response. So for someone who's interested in fear behaviors, zebrafish are just like the oasis of fear behaviors, apparently. So this is a zebrafish. They are wonderful creatures. They're fantastic to work with because they've been really well documented and studied. And so this is why we chose to work with these animals. To give you an idea on size-wise, they're very small. And so when we compare that to a human, their brains are approximately 27 times smaller than theirs. So just think about how you're going to work with that, because it's really difficult. So as fun as continually tapping the glass of the aquarium is, that doesn't tell us a lot about fear behaviors. So in order to do that, we have to take the brain out of the fish and look deep inside for a region called the amygdala, which is central for fear responses across organisms. So here we are trying to, oh wow, you can't see that. That's fine. So we take the brain out of the animal and we put it in a thing of paraffin wax that is supposed to be visible but is not. And we slice it really thin on a machine that's very similar to a deli slicer. We're not actually using a deli slicer, but it's very similar. And this allows us to get really deep inside of the brain to this region that we're interested in. So yeah, that sounds cool. In case you're confused, we need the brains first. So this is really where a lot of the problems that I encountered come from. The only way I can begin to explain this problem to you, I think it's really well demonstrated from this clip from The Office.
Hold on. Judges in session. What is the problem here? You put my stuff in jello again. That's real professional. All right, I do too. It's my steak. No. Take it out. You have to eat it out. Because we're starving people.
Okay, so from this video, we can see a few things. Okay, so we have a stapler and jello. When Dwight realizes his stapler is in jello, there's a few things that seems obvious, but hang on with me. You can see the jello and you can see the stapler. There's obviously a difference between them. But one of the crucial differences between these is the differences in surface texture. So we have a hard plastic surface for the stapler and we have gross jello for the jello. So when Dwight goes to reach his hand in, he can obviously feel a difference. I know this seems very obvious, but this is a crucial part of this experiment. When you're doing dissections, it's important to be able to feel the different textures between one another, and you can do that with the jello. Now, when we work with zebrafish embryos and young zebrafish pictured here, we don't have a lot of those luxuries, that we can't see a difference between the brain and the skull sometimes, and let alone feel a difference. They're very underdeveloped, and so that part of their body just isn't fully done yet. So again, I'm going to try and explain this to you in jello terms. So here is our jello skull, represented by this red blob of jello. And our jello brain, or our brain, oh, we lost the jello brain. Okay, well, there was a jello, okay, yep, there was a blue blob of jello. So if we had a blue blob of jello in there, imagine trying to pull them out from one another using tweezers. You don't have the surface texture to tell you, yep, that's brain, don't jab that. And you just, it becomes a mess. And this is a lot what my summer research projects look like, was just like, what's skull, what's brain, I don't know. It's just really mixed up within each other. And you're probably thinking, wow, that seems like the most crucial part of the experiment would to be get like the good brains out. And you're right, like that was like the most important part, and that was the part that I kept messing up time and time again. But this was me all summer.
I am slowly going crazy.
Yeah, okay. So, yep, that goes on for lots of minutes if you're interested. So what are some things that I learned? One, it's incredibly easy to impale brains with tweezers. Like lots of jabs in different directions. I also learned that it's really easy to cut brains in half with scissors. You can use that with dissection tools as well. I don't think you understand all of the different directions you can cut brains, like hot dog style, hamburger style, like diagonal, like any way you could imagine. I learned how to do that. It's a really great skill. And then finally, it's really easy to leave fish head parts on the brain. And again, this seems like intuitive, but it's super easy to leave like skull and eyeballs and like scales. It's gross. And again, here's a great song. I don't know if... Lots of fish heads. Okay, this is also something else I've learned, is when I was preparing this presentation, I learned a lot of people don't know this song. This is a song I listened to as a child. So if that gives you why I'm interested in fish heads, here we go. So to put the pieces together, when we have holes in the brain, we can't study them. Like, that compromises the whole experiment. So again, this was a lot of my summer research. So this is a graphic representation of all of the poor zebrafish that went into my study to really get 3 or 4 really good slices. We needed to learn a lot about how to dissect a lot of different brains. So this is my research. Great. So overall, what are the things that I learned from this? One, I learned a lot about the intricacies and the capabilities of the brain. While we were not working with live zebrafish, just a little puncture to their brain compromises the whole system. Like, that is how complex this organ is and how really great of a system it is. I also learned how to look at complex questions through simple organisms. So we're looking at complex fear behaviors through a little fish that gets scared when you tap on the aquarium jar. And that can tell us a lot about our own human behaviors and how we respond to stimuli. And then finally, I learned I wanted to be a neuroscientist. Yay, all right? So really, for me, as an undergraduate research, this was a really fundamental piece in my undergraduate career that put some pieces in place that allowed me to be like, yeah, I like impaling brains. Like, yeah, let me keep working on that. I want to keep trying. So that is all I have for you. I'd be happy to take any questions.